Okay. So, good evening, everyone. Welcome to the next session for our course on genes, genomes, and the epigenome. So, as you might remember, we will first go over a recap of the topics that we had discussed in the previous class, where we completed our topics on next generation sequencing techniques and then moved on to the next subsection on genes, on the genome, exome, and the transcriptome. So, just for a very quick recap, we have already covered these initial few topics, which includes gene structure, function, and regulation, followed by mutations, polymorphisms, and functional genomics. Introduction to sequencing methods, which included a comparison between Sanger sequencing techniques and NGS, unraveling the genome, exome, and the transcriptome, and currently we have begun this topic and we'll discuss more about this in today's class. This will continue probably only till the next class, and then we'll move on to analysis of high throughput sequencing data. So just for a quick recap of the topics we've already covered, we had initially discussed the Illumina sequencing method, which involved the bridge amplification technology. And under the purview of that, just a second, there's some issue, just a minute. Yeah. So next we had moved on to the sequencing by ligation methods, which are used by Illumina and under this Alexa technology. And then we had moved on to sequencing by synthesis as part of the previous class. So this involved the addition of individual nucleotides, which is single nucleotide addition approaches. So that included the 454 pyro sequencing method, under which actually involved an initial step of bead-based template enrichment using the emulsion PCR method, like you can see here, where you have your sequences of interest or the template sequences which are ligated with the adapters, and along with that we have these microbeads which are then subjected to droplet generation, and within that droplet you also have the enzyme which will an enzyme cocktail which will carry out the amplification or the PCR step, and following that you will have a cohort or a population of beads that are enriched for that particular template sequence of your interest, and then those can be subjected to sequencing. So we discussed two methods under that, the 454 pyro sequencing method, and how it exactly or how the addition is actually recognized was appreciated by this method, where every time an individual DNTP gets incorporated, the release of a pyrophosphate or the PPI group allows the conversion of APS to ATP, and then that ATP is utilized by the luciferase enzyme to produce oxyluciferin, which is then detected by the detector, and that is recognized as the signal for the incorporation of the nucleotide that we are currently looking at. So in subsequent cycles, every time a nucleotide or a NTP, DNTP is incorporated, yeah, every time an DNTP is incorporated, you will be able to read the signal for the same. And if multiple DNTPs get incorporated as well, that leads to a significantly more amplified signal, and therefore it helps us to decode the entire sequence by this strategy. Following which, we also discussed the ion torrent method, which involved the presence of sort of a voltmeter which was able to detect the incorporation of individual bases by the shift in the pH, which occurs due to the release of an H plus ions every time a single base gets incorporated, and that results in a 0.02 unit change in the pH, which is detected by the complementary metal oxide semiconductor and is fed device, and unincorporated bases are then washed away, and every time you get an incorporation, the signal can be read out in the following manner. And similar to what we discussed for the luciferase strategy as well, you can see that when multiple bases or multiple when a tandem sequence is composed of the same base, then the H plus ion release can also be detected, and that leads to a more amplified signal. Following this, we looked at a comparison between short read and long read sequencing technologies, where short read technologies are usually more relevant when you want more comprehensive overview of an entire set of regions that are to be sequenced, which means that you want more information at one glance. And long read sequencing makes more sense when your objective is to know the intricacies of a short region, but in a lot of detail. So this was a contrast between the two techniques. I'm not going to go over the same because we've already discussed this. Following which, we then moved on to real-time and synthetic long-read sequencing approaches, where the smart bell template was utilized, and under that you had two hairpin adapters which were present on either end of the template sequence of our interest, which generated a continuous region that could be, sequenced in a loop, and within that, it sort of gives you a specialized type of a sequence that can be read or that can be simultaneously sequenced at both ends in a continuous manner. And this is present within a specific type of well, which is called as a ZMW well, which are called zero mode waveguides. And within that, you have these DNA molecules or the templates immobilized along with a polymerase, which is then able to incorporate. And every time a nucleotide is incorporated into this sequence, the camera records what sort of a light is emitted, by which you will be able to decode the sequence. So this we had also discussed in a lot of detail, and then we talked about synthetic long read sequencing. Again, keep in mind this was real time long read sequencing, whereas the synthetic one, as the name suggests, has some difference with the previous one. So it actually involves um, sort of using the short read sequencing technique, but for long read applications. So majorly this strategy can be utilized by, or has been utilized by the Illumina sequencing method and by 10x Genomics as well, whereby they actually fragment DNA and they barcode that region, and then the barcoding is carried out in a manner where the same within the same well, all of the DNA sequences, they carry the same barcodes, and then they can be traced back. But what you're actually doing is you are dividing a particular region into much smaller sequences, which can then be read in a lot of intricate depth. So this is overall the strategy for synthetic long read sequencing as well, with which we had completed our discussion on the NGS methods. And then we had moved on to the topic on unraveling the genes, the exome, or the genome, the exome, and the transcriptome. And under the purview of this, we had started an initial discussion where we understood the contrast between whole genome sequencing, whole exome sequencing, and targeted region sequencing. So I'll not go over the details of this. Again, it's very similar, and we'll be taking up WES or whole exome sequencing in a lot of detail in today's class. Reason being, first of all, it is the most commonly utilized. We have already discussed why it is more practically relevant, and also because there's one thing that I want to correct from the previous class where I was asked about how the exome exactly is captured, and under that there was some confusion that where the exome capture strategy was not very clear, and instead it was accidentally proposed to be similar to how qPCR is carried out. So we'll rectify that in today's class. My apologies for this name, but we will be discussing all of these steps. Like here you just see a step where fragments are generated and you get reads. So what happens between all of these or within all of these steps, and what is the amount of effort, the resources, and what actually justifies the cost of this technique. We'll discuss all of that in a lot more detail. But overall, what I want you to just keep in your mind for now is that WES or whole exome sequencing is more translationally relevant in a lot of manners, or why it is more practically utilized is because you are sequencing the coding regions of the genome, right? Now, the coding region, please keep in mind, does not necessarily mean only the exonic regions. It also includes some other regions, which we'll talk about later on. Secondly, it has a lot more depth because its minimum sequencing depth is of 50 to 100x. Nowadays, you get up to 200x as well. So that means you have just to put it into a layman's language, you have at least 200 reads which are supporting the sequence for a particular region. So the confidence or the accuracy of this technique is a lot more improved. In contrast, because the genome is a lot larger, the amount of information that you can, that computers at a time or that is cost effectively justifiable is is a little more challenging. So therefore, the depth for whole genome sequencing is usually only 30x, but also keep in mind that there are no gaps in the information which is getting sequenced. So it is a lot easier to align to the reference genome and therefore is a lot more reliable. But yes, the amount of information that gets sequenced in this case is a lot more, and therefore its cost is immense, and the amount of data which is obtained as the result or the output of whole genome sequencing is also extremely huge. So with all this in mind, we will move forward with today's session. We are currently in week 6. Okay, I need to correct this. But we're currently in week 6 and we are going to discuss topics under the purview of this subheading, which is genome, exome, and transcriptome. We'll talk about the exome and its sequencing approach. We'll talk about the key steps in the sequencing protocol, and we'll discuss some of the expected outcomes very briefly because we'll have a lot more detail about this in the session where we talk about how high throughput sequencing data is actually analyzed. So we'll do that in a few more classes. So, first of all, beginning with the key concept, which is about understanding the exome and the whole exome sequencing technique. So, what defines an exome? An exome is the sequence of all the exons in a genome, reflecting the protein coding portion of a genome. So, that is the simple definition of an exome. In humans, the composition is approximately 1.5% of the total genome. Now, like to this is how we had basically discussed it in the previous class as well, that the collection of all exons forms the exome. But it also includes something else, which we are going to stress upon today. So it also includes elements that are not protein encoding, like sequences of the microRNA or long non-coding RNA. So this is some additional information that you also get from the output of whole exome sequencing data. And therefore, this is another sort of analysis that can also be carried out. And of course, you want to maximize the output that you can get from one sequencing project because these are extremely expensive. So with this in mind, whole exome sequencing, or WES, represents a genomic analysis technique that is geared towards the detailed investigation of all transcribable exons within a genome. How is this accomplished? It is accomplished by leveraging DNA microarrays or whole genome sequencing platforms in tandem with sequence capture technologies. This is what we'll discuss in more detail today. So, as the name suggests, it is basically an analysis technique or a high throughput sequencing technique where you want to get an idea of all the transcribable exons, or rather, I think a better way to read it is the information from all of the transcribable regions within a genome. So how you do that is either you use DNA microarrays, which has become somewhat more a little obsolete in today's day and age, but it is still performed. Or what is usually opted for is the high throughput sequencing method, and that involves also sequence capture technologies, which minimizes the amount of data, which will or minimizes the amount of information that needs to be decoded or sequenced, and also makes it practically a lot more feasible. So these tools, they basically allow for the selective capture and enrichment of exonic regions in the DNA, which are then subject to high throughput sequencing for insights into gene expression regulatory mechanisms. So you're subjecting these. So what you're eventually doing is you're using high throughput sequencing methods using all of these strategies we have discussed previously, like sequencing by synthesis, sequencing by ligation, all of the different companies that we talked about, like Illumina, for example, or 454 pyrosequencing by Roche or Agilent, all of these different companies. And we are then getting more information out of it. Now, WES exhibits the capacity to detect potential mutations that may be associated with phenotypes or diseases, as well as overall genomic variations, novel gene discovery, and the phenotype to genotype relationships of gene mutations. The exome, therefore, what you're exactly getting out of this sequencing project is that you can detect any coding or any change in the coding sequence of an individual. So that makes it an excellent technology to detect mutations that can, affect phenotypes or diseases. You can get an idea of genomic variations. You can actually see the phenotype to genotype relationships of gene mutations. So not all mutations are coding, or not all mutations will actually have an effect on an individual, but something which is present within the exonic region or within the coding region can actually be significant, and we are able to capture that. So that is the practical utility of using this method. Moving forward, how is it different, or what segregates whole exome from whole genome sequencing projects? So, as we have already discussed this previously, I still want to, stress upon it because eventually, if you are considering moving forward in this direction into learning how to analyze these data, and if you want to use this, and actually it's become almost indispensable in today's time, where utilizing sequencing, high throughput sequencing technologies is very important to get an idea about what are the altered pathways and what can actually drive the onset of a disease. So, taking that into consideration, knowing what distinguishes these techniques and what is relevant to the project you are pursuing can become very important. So, broadly speaking, what is the difference between WES and WGS? WES, or whole exome sequencing, entails the extraction of all exonic regions. Please keep this in mind. What do we mean by this? You need to extract exonic regions. It's not that only exons will be present within any individual, but you want to extract all of them. The prime dominions orchestrating protein synthesis. Now, what do these proteins do? They form the structural and functional core of the human body, rendering the exome zones within DNA of greatest genetic value. Given that exons constitute merely 1% of the individual's genome, WES successfully manages to control costs while it amplifies testing, the depth of testing, which means it increases this sort of confidence interval, or increases the confidence on your sequencing accuracy, and makes the results a lot more reliable. Significant academic research outputs are currently centered on the exonic region, and therefore data which is generated from WES is adequately substantial for future updates. With the advent of fresh research findings, direct data analysis can be employed without necessitating a retest. So this is what whole exome sequencing looks like. So you have all exons of all known genes, which is only about 1.5 to 2% of human DNA. You have variable read depth at boundaries. You get a lot more sequencing depth, and you make it a lot more cost effective. In contrast, when we talk about whole genome sequencing, as the name suggests, of course, its objective is to capture all genetic loci. Areas beyond exons, which account for 99% of total loci, contain abundant repetitive and nonsensical elements. Now, please don't interpret this in any other manner. When we say nonsensical element, it doesn't mean that it doesn't have any role. Of course, it has some regulatory role. It gives the opportunity for the genome to undergo real-time editing, and by editing, I mean the fact that it can have sort of changes which make it like how do I explain it? Like for example, splice variants or the incorporation of fusions in a lot of cases like gene fusions or some opportunity for rearrangements within the genome, which can have significant impact on the coding protein and effectively upon disease onset. All of that is subject to um, alteration. So it sort of increases the dynamicity of the genome. Dynamicity means it makes the genome a lot more prone to change, which again fundamentally the concept we have about the genome is that it is constant, but that's not actually the case. It is um, maintained, but it is not constant. It is subject to change based on the type of regulatory elements which are controlling its expression. uh, the yield of relevant scholarly literature is also markedly skimpy, implying that the legible content, which is derived from WGS, is nearly indistinguishable from WES extracted content. Now, don't take this otherwise. What do we mean by this? The legible content is not distinguishable. What we mean by it is that usually, whenever you want to study any disease, again, I'll take the example which I personally am most familiar with, and what you'll find very commonly in academia and research as well, is that these sequencing technologies are very important for diseases like cancer and many different types of cancers. Of course, they're used for other diseases as well, but cancer is something which is very heavily studied using these techniques also. So most of the time, changes that you will see within the exonic region or within the coding regions of the genome are actually what will make a lot more or what will have a lot more impact on the disease onset in comparison to any change which occurs within these intergenic regions or intronic sites or non-coding sites within the genome. So, taking that into consideration, the effective cumulative information that you get from uh, WES and from WGS effectively can wind up to be the same, or at least to an extent similar. However, it is not correct to say that the that the information you get from whole genome sequencing is absolutely irrelevant and is it's a waste of money or anything of that sort. No, not at all. One of the major subsets of information that is completely missed out on or is not very reliable when we talk about whole exome sequencing is getting information on the uh, structural variants. Why is that important? It's because in cancer like diseases or in a subset of cancers, genomic structural rearrangements, and when I say rearrangements, it includes a subset like um, reciprocal fusions or large insertions, deletions, or information about transposons. All of that information in a lot of cases can be misrepresented or misinterpreted when you use data derived from whole exome sequencing because it is leaving a lot of gaps, as you can see here. So, for example, if some fusion has occurred within this region, of course, currently the tools that are available like fusion culture, etc., they will be able to detect such fusions, but the reliability of those techniques and the practical applicability of these techniques can be limiting in a large number of cases. And therefore, having continuous stitched information, continuous um, reads for a particular region will make the description of these structural variants way more reliable, and therefore WGS is more important when you want to see such intricate changes within the genome. Apart from that, the entire human DNA or the entire genome can be sequenced, including introns, RNA, genes, etc. All of that is an advantage. The read depth in this case gets significantly compromised. Obviously, we have discussed this before that read depth can go up to 30x. So there are only 30 reads which are supporting the sequence of a certain region. But then where do you get confidence from? You will have a lot more confidence because they can all be stitched together and the interrupting regions are a lot less. Similar read depth across the genome can identify copy number variants, repeat expansions, but the cost is much, much, much higher. Additionally, in context of this cost itself, significant costs are disproportionately disproportionately squandered on pointless loci in whole genome sequencing, which makes it very challenging to guarantee sequencing depth. In certain WGS genetic tests and that cost several $1,000, the average depth is a mere 30x. For the loci under analysis, the sequencing depth could dwindle to single figure levels, rendering the data practically valueless for reference. Now, again, please keep in mind that this was the source that I took it from is trying to stress upon the advantage of whole exome sequencing, and therefore it has been a little harsh with the usage of WGS, but it is not a completely useless technique, of course not. It's just that the contrast between them is extremely stark, and what you find here is a lot more practically applicable. But for academia, whole genome sequencing is what you'll find in a large number of papers, and the amount of information that can be decoded from there is also immense. With this knowledge, we'll move forward and understand the key steps that dominate or that construct or that formulate the principle of whole exome sequencing or WES. Whole exome sequencing, like we've already discussed, is a genomic analysis method or a genomic analytical technique that seeks genetic mutations tied to protein function variations. What does this sentence mean? It means you are analyzing the genome, which means you are seeking genetic information, changes or alterations within the genome. But why are you seeking that? Because you are trying to connect that information with variations in protein function. Not protein structure, protein function, because you see some sort of a phenotype, some sort of an abnormality in protein function in an individual with a certain disease. You want to connect it to the information that has undergone some change in the genome, in the genetic sequence of that particular individual. So the basic principle encompasses a couple of steps. What are they? First is DNA capture and enrichment. DNA or RNA probes, which are specific to exon regions, are used to capture and enrich for those DNA sequences. Don't worry, we'll discuss all of this in more detail. But this is what I wanted to stress on most significantly at the beginning of this class, that contrary to what I had discussed in the previous class, please note that it is actually the use of probes and hybridization based strategies to enrich for exonic regions for high throughput sequencing. And what I discussed as the conversion of RNA to DNA is the strategy used for qPCR. I misspoke at that point of time. So please correct this in your own dictionary as well. So typically, how is this done? This is achieved through liquid phase hybrid capture technology, which employs the principles of base pairing to hybridize biotin labeled RNA probes with DNA libraries with adapter sequences and then enriches the DNA of the target area through magnetic bead binding. What did we say? A lot of jargon came here. First, you use some probes, which are either DNA probes or RNA probes, to exon regions, which are used to capture and enrich for some DNA sequences, which form the exome. Typically, how is this done? You use some sort of a hybrid liquid phase hybrid capture technology, via which you hybridize these biotin labeled RNA probes with DNA libraries with adapter sequences, and then you enrich for the DNA of the target area through magnetic bead binding. You use some magnetic beads and you separate out those regions. Second, you perform high throughput sequencing. The enriched DNA sequences, which are now selected for, can be sequenced through high throughput sequencing technology, which is the process of discovering all the DNTPs and the arrangement of the DNTPs relative to each other in the exome with respect to a particular position within the genome, which helps us to understand potential pathophysiological changes in certain diseases. Third, of course, is data analysis, whereby the sequencing results are subjected to bioinformatic analysis to pinpoint genetic mutations that are associated with protein function variations. So the conventional function The conventional working process of whole exome sequencing involves a couple of steps, all of which we'll discuss in detail in the upcoming slides. Overall, what we'll be discussing in more detail today is this DNA capture and enrichment step, because high throughput sequencing and the different strategies of high throughput sequencing are topics we have already covered in the past couple of classes, the past, I think, 3 classes or so. So we'll not go into details about it. Today, we'll cover whole exome sequencing, DNA capture and enrichment steps. And in the next class, maybe we'll start off with a little bit detail about RNA library preparation and whole genome library preparation. And then we'll move on to data analysis and the tools which are used for data analysis in the subsequent class. The conventional working process of whole exome sequencing, it involves a couple of common steps. What are they? First, you have sample preparation, you have exome capture and library design, followed by target enrichment, sequencing, and bioinformatic analysis. Again, this is kind of recapitulating what you've already seen here. Yeah. Under, just a sec. Yeah. Overall, the flow chart here represents how whole exome sequencing is performed. So under sample preparation, we have first DNA extraction, its purification, and quality control, after which sample can be preserved. That is, once it has passed QC or quality control, a sample has been sort of approved for sequencing, and therefore it can be preserved at this step. Any sample which has not passed QC will usually either not be allowed to proceed for sequencing, or it will be sequenced at the students or at the customer's request. So whenever you get some sequencing done, if you send suppose 50 samples, all 50 samples will be checked. They'll be investigated for the quality of the DNA or RNA which has been isolated, for the integrity of the molecule which you're sequencing, and only those that have qualified this check will be allowed to pass, and all the others will be sort of sent to you for your approval, and only then they will be allowed to proceed for sequencing. Because of course it is an um, like if your sample is somewhat compromised, say for example, this most commonly happens with transcriptome sequencing or RNA sequencing. Why so? Again, like we've already discussed, DNA is a lot more stable. So whenever you are shipping some sort of a sample to, say for example, somewhere outside your country or to some other destination, which is, which has the company that is an expert at this sequencing method, then a lot of times what happens is that the sample can actually get compromised whenever it is RNA, because RNA is very prone to degradation. It is the easiest thing to degrade RNA because it is so ready to do so. Therefore, to prevent that, you have to maintain certain conditions, you have to maintain the temperature conditions, you have to optimize the shipment or optimize the time that it takes for the shipment to be delivered, so on and so forth. So in certain conditions, it might so happen that the sample has actually degraded, and therefore it might not actually pass the quality control step, and therefore you should ideally not proceed for sequencing with such a sample. But if it is something that is extremely crucial and you do not have any backup or you do not have any alternatives to it, you can proceed for it. But again, its results will not be very reliable. And honestly, whenever you want to publish the data which is derived from such a sample, you will have to provide the quality control report, and then it will get severely scrutinized by the reviewers of your manuscript, and then it becomes very difficult to defend such a sample, and you should actually ideally not proceed with it. So yeah, so that is the quality control step, what I just mentioned, following which we actually proceed with DNA fragmentation. Again, if you remember, we have discussed shotgun sequencing a lot of times and have understood the significance of it and the methods which allow for shotgun sequencing to be performed. So under that, DNA fragmentation is the most crucial step. And following that, you have exome capture and washing and elution if necessary, which allows for target enrichment. And this can be repeated multiple times to enrich for your template, to enrich for your target, which will be used for sequencing. Yeah. Following this, we also have the third step, which is actually the sequencing step. And under the sequencing step, we of course have to perform high throughput sequencing of the region which has been captured, which in this particular scenario is the exome. And then data analysis can be performed using the same. Hmm. Yeah. Now we'll discuss these steps in a lot more detail. So first and foremost, the key steps in whole exome sequencing. First is sample preparation. Yeah. So sample preparation, what kind of samples can you actually send for sequencing, or what can be used for sequencing? Again, another thing to keep in mind, usually for most companies, both options are available. Either you can ship your tissue that you want, or you can ship the biological sample that you want sequenced, or you can also send the isolated RNA or isolated DNA, whatever you are getting sequenced, to the company. Now, that depends on how crucial your sample is, how indispensable it is. Most cases, it will cost a lot lesser to isolate the molecule you want sequenced and then ship it. But of course, it will also be subject to the challenge that the sample might get ruined. But it's usually a better policy to isolate the macromolecule yourself to avoid any chance events, because companies might not, prioritize that as much as you would. So yeah, it depends upon the context and the facility that you have in your lab and the financial backing you have for your project. But commonly for whole exome sequencing, the kind of samples that can be usually utilized include these following, which includes whole blood sample. You can use PBMCs, or the peripheral blood mononuclear cells, freshly frozen tissues, which are basically fresh tissues that have been frozen. You can use those. Following that, you can use FFPE, or formalin fixed paraffin embedded, which is FFPE samples, which are usually used for staining purposes, because one of the reasons it's not the most ideal sample to use for sequencing, especially because you're investing so much of because so much financial investment goes into these sequencing methods, is because FFP samples after formalin fixation, they are prone to certain mutations becoming more prevalent. I think it was a T2A or A to T conversion. I will check it once and then get back to you. But basically, it's not always very trustworthy to go for the data from FFP samples, and it is much more appreciated and much more reliable when it is derived from fresh frozen tissue or from blood cells, because they have not been fixed in any manner, they have not undergone any changes, and it reduces the chance of error and increases the reliability of the sequencing strategy that you're using. Um, Apart from that, you can also use plasma samples, you can use amniotic fluid cells, you can use, of course, amniotic fluid is, cells from amniotic fluid are usually going to be utilized whenever you are doing some sort of genetic testing for for the patient under consideration or for the expectant mother. Along with that, you can also use any other type of basically cells from which you can extract the DNA. And the more the number of cells that are available for that particular patient, the more amount of starting sample and the more depth you can actually opt for, and the more reliability you can actually have. So again, it is a very context specific thing, and the tissue type and the type of sample you're analyzing, all of that also makes a lot of difference. Now, what do you do? Suppose you have a patient's biopsy sample from a cancerous site, and you want to get it sequenced. What do you do? So DNA needs to be extracted from the biological samples that you are trying to check for or that you're testing. And then it has to be subjected to fragmentation, again, similar to what we have already discussed for the shotgun sequencing method. And then it can be subjected to exome sequencing. Now, DNA is extracted, and this extraction part, again, it depends upon what type of facility you're paying for, basically, to the company that is doing the sequencing for you. Also, another thing to keep in mind is that whenever you are reporting any sort of these high throughput sequencing data, one very crucial point to keep in mind is that you should always have a control. So for each individual, suppose you are trying to check for mutations which are present in 50 cancer patients, 50 breast cancer patients, say for example. If your objective is to check for that, then for each of those 50 patients, you should have some sort of a control sample. Most commonly, what is used is the blood. Why? Because many a time, some patients might have some um, alterations that are not actually going to that have not been acquired by the patient in their adulthood, but they're actually present within the patient from a very young age, or by birth. What are those? They are germline mutations, which are part of the germ cell that they actually arose from. Now, germline mutations also are relevant in some contexts. Like, for example, if a certain population or if a certain subpopulation of individuals from a certain region are more prone to a disease, then many a times germline mutations also need to be studied. I'll give you an example of this. Say, for example, the disease that I personally work on, which is prostate cancer, is extremely prevalent in the African population and has been prevalent for a very long time. And there has been some speculation about the ancestry or about the African ancestry having some proneness to this particular disease. So, if you want to actually check that, what you need to do, you should have the sequence of their germline DNA as well. And then you need to check whether the germline itself carries some sort of a base change or some sort of an alteration which is making them more prone or more predisposed to suffering from the disease. So that is the information you can get from the blood sample or from any germline sample. In contrast, for most diseases where mutations are not usually not inherited, but instead they are acquired by an individual over time, and that acquisition can often be attributed to, some sort of some sort of an agent which is causing the disease. Example again, let's take the example of cancer itself. So many a times cancer is actually, or actually most of the time, cancer is the result of some somatic mutations that have been acquired by that particular cell over a period of time. You can learn a lot more about this from one of my previous courses on the ISSS portal, which is on advanced concepts in cancer biology and precision medicine, where I've discussed this in a lot more detail about acquiring genetic mutations and the effect of those genetic mutations over time. And yeah, and apart from that, what we also see is that the information from the germline DNA also helps us get rid of any germline mutations and only focus our attention on the somatic mutations which have arisen from somatic cells that are present in the adult body, and therefore they have a lot more. The possibility of them affecting the disease onset and disease progression is a lot more. Yeah. So let us again focus our attention back to the text at hand. So, physical disruption using shearing, sonication, fluid dynamic shearing, where they serve as primary methods of DNA fragmentation, can often be used or are often used. One of the most common methods that like we use in the lab ourselves, and something we'll be discussing also in today's class, involves the use of an instrument known as covarice, which is sort of like a sonicator, but a very sophisticated sonicator, which allows you to adjust the settings in a way like you can adjust the duty factor, you can adjust the RPM and a few other parameters such that you can actually instruct the instrument that this is the thickness or the length of the DNA that I want it to be fragmented to. So, for example, when we perform chromatin immunoprecipitation, we usually go for 500 or 700 base pair long sequences, but here you can adjust it to get a certain length like 150 base pairs or 200 base pairs or 100 base pairs and so on and so forth, so that much smaller regions can actually be subjected to sequencing. Um, yeah, that is the physical disruption method, including shearing of DNA or sonication of DNA. The capture and the enrichment of exons. So during exome capture and enrichment, exons get selectively enriched using the exon or the exome capture technology. The fundamental concept behind target enrichment pertains to exploiting the physicochemical differences between the target compound and other substances to facilitate their separation. Following the isolation of the exome from the rest of the genome, you perform multiple washing steps to refine or to obtain a pure set of only the coding regions of the genomes or only the exome of the particular cell or of the population of exomes for a particular sample. Although distilled water is typically used to elute the target, sometimes certain specialized kits which are used for exome capture might also necessitate some specific elution solution. All of that depends upon what you're using, what is the principle that the kit follows, what is the strategy, which company is using which strategy, a lot of different parameters which we can kind of skip for now. But basically, you want to enrich for only the exons while getting rid of um, any other nucleotide sequences for your sample that you are using for the sequencing project. Moving forward, the step 3 in this case is construction of a library. What does this mean? Let me just focus here. Yeah. So library construction actually involves repairing the ends of enriched exonic DNA fragments, attaching connectors or adapters, amplifying the libraries to convert the enriched exonic DNA fragments into sequencing libraries so as to provide ample DNA template for subsequent sequencing. Don't worry about this, we'll discuss it in a lot of detail today. Basically, what I wanted you to see here is that there are a lot of different kits that are available in the market for target enrichment for whole exome sequencing. The Agilent Sure Select, for example, this is most commonly used. You also have Agilent Sure Select XT2, V6, V5, IDT's exome panel. You have Illumina's Nextera, Illumina's TrueSec also. I'll be discussing Illumina TrueSec today. And you also have Roche NimbleGen exome capture kit. And you can see that the region which can be targeted using these slightly vary among different methods. And you can see that the amount of genomic DNA which is needed is also different. Among these, Illumina Nextera is the most sensitive. It only needs around 50 nanograms of genomic DNA, and that's good enough. But most other strategies use 100 nanograms. No. Yeah. So you also have the adapter ligation step, which is for most cases, you can see that ligation is what is the ligase or the ligation step is what allows for attaching the connectors or the adapters. For Illumina Nextera, you use transposase for this purpose. You can read about it, or maybe we can discuss in the next class. Today I don't have it, but tagmentation step actually allows for attachment of these specific sequences. You also have to focus on the probe length, where the length of the probe can also be extremely variable among these techniques, which is, of course, undergoing constant changes and constant development to improve each of these methods so that you can also check in real time. Now we will, in continuation with what we have seen here, that initially sample has to be prepared, following which you have to capture the exons and enrich them. Then you need to construct a library out of it, for which you will like you generate a or you enrich the population in a certain manner for these exonic DNA fragments and then use them as templates for subsequent sequencing. Then in step 4, you actually carry out the sequencing that we've been talking about for so long, which is, of course, which involves a lot of different steps. Like, for example, you have the magnetic beads and Roche 454, like we just discussed, the pyrosequencing method, the Illumina HiSeq, bridge amplification method, sequencing by synthesis, sequencing by ligation, all of those steps. The rationale behind NGS involves coupling the exome sample to an appropriate base, such as a flow cell in Illumina HiSeq and magnetic beads in Roche 454, followed by an in situ PCR duplication to ensure signal amplification in every round. The DDNTP is examined after each round of extension, and the complete sequence is eventually compiled using an algorithm. Don't worry about all of this. We'll discuss everything when we actually come to the analysis part. The high efficiency and the high throughput capabilities. So, the high throughput capabilities render NGS as an appealing option for high throughput sequencing method. Finally, as part of data analysis, what do you first do? You eliminate any low quality reads during this sequencing process through quality control, which is FastQC. Then you align high quality reads with the reference genome, quantify SNPs or single nucleotide polymorphisms, insertions, deletions, copy number variations, followed by annotation, screening, analysis, and validation. Data analysis constitutes a critical juncture in exome sequencing, encompassing the evaluation and processing of sequencing data quality, aligning sequence reads to the reference genome, and identifying genetic mutations within the samples. The detected genetic mutations need to be annotated, including discerning the function of the mutation, its pathological significance, and its potential impact. So, all of that information can be decoded later on. Now, overall summary of the steps which are carried out, after which we'll take up the library preparation in a lot more detail. First, you have to align the reads, which means you have to map the reads that you get as output of the whole exome sequencing project. Those reads need to be mapped to the reference genome, which means that they basically need to be annotated, or they need to be decoded to see where they have come from. um, and you have to process it, you have to improve alignment, you have to mark PCR duplicates, following which you have to do variant identification, which means that you have to check for any sequences that are very variable across multiple reads to figure out that yes, this is actually a true variant and it is not just a sequencing artifact or a PCR polymerase error or anything of that sort. And then you have to do a genotyping based on that. Then you have to remove any false positive or likely false positive variants. You have to filter those out, and then you need to visualize and perform downstream analysis. You need to do in vitro validation and so on and so forth. Yeah. Now we will discuss exome capture and enrichment in a lot more detail and then take up each of these individual steps. Overall, you can refer to the Illumina TrueSec library manual for your for more knowledge and for better insights into this in a lot more detail. But today we'll be going through the summarized steps for our understanding for today's purpose. So the preparation of an exome enrichment library follows DNA extraction. After you have extracted DNA from whatever sample you have taken up, whether it is patient blood, PBMCs, patient biopsy, freshly frozen tissue, or it is FFP sample, or whatever it might be, then you have to subject these samples or subject the DNA isolated from those samples to exome capture and enrichment and library preparation. Agilent, Illumina, and Limbelgen under Roche are three very commonly utilized exome capture kits. They've been compared and summarized in the next slide, which I'll show you guys soon. Then, product selection should be influenced by platform specific strengths and weaknesses. Most commonly, people use either Agilent or Illumina. Today, we'll be discussing the Illumina method in a lot more detail because that's what we've been following for so long. You can read about all of them. There are, of course, some differences between each of these strategies, but it's not possible to cover every company's uh, in IP in in one or two classes. So I want to just expand one technique in a lot more detail, and the rest can be knowledge is endless. So you can read up as much as you want. Today we'll be discussing Illumina's true sex strategy. But despite all of these differences, all these capture technologies, they obey the same three basic principles. First is DNA fragmentation, second is adapter ligation, and third is target enrichment. Now, what are the key steps? First, of course, the genomic DNA or the gDNA gets sheared into random fragments. Either it is done mechanically or by using some ultrasonicators like Covaris, like I just previously mentioned, or it can also be done biologically by performing enzymatic digestion. Again, the example for this is the use of transposase enzyme for this purpose. Then the fragment ends are subsequently blunted and ligated with adapters, under which Agilent's Sure Select XQT, QXT, and the Illumina and Xterra platforms use a shearing-free transposase-based library preparation. Targeted enrichment of exonic regions follows library preparation, and the method of a method which is used for this platforms. Finally, Agilent's Sure Select Human All Exon, Nimblegen's SecCap Easy Exome Library, and Illumina's TrueSec, they use a hybridization method with complementary baits and magnetic bead mediated pull down, while Agilent's Haloplex platform uses an amplicon-based capture. Non-targeted sequences are washed away, hybridized fragments are eluted, the resultant enriched library is then amplified. Basically, what do we mean to say? We mean that you start off with a lot of information. You start off with the complete DNA sequence. From there, you fragment it into small pieces. From there, you use some baits. Bait basically means something which can um, filter out or which can attract the prey, the sequence of your interest, which is your exon, and using this, you then immobilize it onto a magnetic bead. Then you pull down the magnetic bead or you separate out the magnetic bead, which helps you specifically select out those sequences which are of your interest. And that gives you the template which you can use for sequencing. In contrast, Agilent's Haloplex platform uses an amplicon-based capture. Now, non-target sequences, in this case, they will be washed away, they'll be gotten rid of, and what you actually need will be then amplified. So now we look at the steps which are a part of this entire TrueSec library preparation conducted by Illumina in more detail. First, let us look at the steps. Usually, this is the strategy which is utilized. Again, there are ifs and buts to this technique. Please make sure that you understand the subtleties between them. It's just one strategy we are using or we are discussing in more detail as a representative example. There are differences between different methods used by different platforms. So for the TrueSec library, on day one, first what is done, you fragment DNA. These are some of the reagents which are used. You can read about them in more detail within the manual. Following which, what you again see, look here that there are some stopping points where you can initiate sample preparation, but take a pause depending upon what is the availability of reagents and resources. But apart from that, there are some steps which are compulsory and that need to be followed. So after DNA fragmentation, you have to perform end repair. You have to select the library size, following which you need to adenylate the 3' ends of the template you are using. Why will come to in the next slide. Then you need to ligate adapters. Again, you can see with the adapters, there are a couple of different reagents also present here. Following which you need to enrich for DNA fragments using some certain specific types of strategies. Then you need to hybridize your probes of interest. You need to capture the probes that have hybridized DNA sequences of our interest. Then you need to perform another round of hybridization for more accuracy and more reliability. You again need to capture these hybridized beads or these hybridized probes, following which you need to clean up non-specific sequences from this library, amplify it, and then clean up the amplified library, which will give you the final panel or the final library which will be used for sequencing using all of these strategies we had discussed previously. Specifically, if it is Illumina we are talking about, it will use the bridge amplification strategy for sequencing. So many steps are present prior to the actual sequencing which needs to be performed. The library preparation step, like we have previously mentioned, that it involves all of these little sort of intermediate steps prior to it. This is what I was talking about as table one, the comparison of different exome capture kits. Like you can see, there are a lot of differences. What we are discussing today is this Illumina TrueSec exome enrichment strategy. But apart from that, there are some platforms by Agilent, by Roche, which is the Nimble Gen 1, and by Illumina Nextera also. Um, you can see that the target size is different. The probe size which is used for each of these methods is different. How many probes are used, that is also showing a lot of variation. Like for example, the Agilent Haloplex exome and the Nimblegen SecCap, they have a lot more probes in comparison to the other strategies. And what these probes actually target, that can also vary. And therefore, whenever you send out your samples for sequencing, you need to read about the regions which are included and excluded as part of these probes and whether or not that aligns with your objectives. Following which, the number of reads on target and what are the percentage of target bases that are covered at more than 10x, and therefore when you go on to 100x, then how much of it is actually covered, what is the input DNA for each of them. Like you can see, for TrueSec, such minute amount of DNA is required. Again, this is very important because many a times when you're using patient tissue samples, like for example, we talked about uh, the amniotic fluid cells and the DNA which is derived from them. Obviously, the number of cells that you can use from there will be very limited, and the amount of information you can get from there is also very limited. Or, for that matter, many a times people actually do the biopsy of or people actually extract cells from a blastula or from a developing from some intermediate developmental stages for some fetus for a fetus. So in that case, the number of cells and the amount of DNA you can have is very minimal, and therefore, depending on what is the amount of input DNA you have available, depending on that, you can choose the platform on which sequencing can be performed. Like, for example, some techniques are more sensitive, some are less. Like the Illumina Nextera is very sensitive. It only requires 50 nanograms of DNA, and therefore it is more useful when you have very limited amount of input sample. Apart from that, the strategy which is used for fragmentation can also vary. And like you can see, for each company, they have one strategy which uses the enzymatic method, and one which, or at least one strategy which uses an enzymatic method. But what is usually more reliable is the use of ultrasonicators for shearing purposes. what are the strengths of each of these techniques? For Haloplex, very little amount of DNA is required. For Agilent Shaw Select, you can see that duplicated reads are very low. For indels, it has very good sensitivity. Indels are insertions and deletions. Um, it has a very high alignment rate, so the variation that you obtain is quite low. Following which you can see for Nimblegen SecCap, you can see it has more uniform coverage. Most reads are on target. Um, for Illumina Nextera, it has the best coverage of UTRs or untranslated regions and microRNAs. So if that is is your focus and that is something you are interested in learning about from your sequencing project, like you have one sample but you want this information to be included as well, then you can opt for Illumina Nextera. It is also a very fast technique. In comparison, you can see that for TrueSeq exome, the coverage there is also very fast, but it does not claim that the speed it performs it at is as high. And of course, its recommended DNA input is also larger and it involves ultrasonic sonication as input also. So that makes it, these are the strengths or the advantages of each of these techniques. Now, when we talk about the weaknesses or the cons of these techniques, then you can see that for Agilent Haloplex, sometimes you can have very different or varying target regions, length of target regions. For Agilent Shaw Select, it is very significantly affected by high GC content, and the efficiency of the sequencing technique in such GC rich regions can actually get compromised or can be quite limited. Fewer reads are on target compared with Nimblegen. For Nimblegen, however, alignment rate is much less than that of Agilent. Then you can see that for Illumina Nextera, it has a large number of off-target reads also, which is the same for Illumina TrueSec as well. For Nextera, coverage bias is present or is observed in GC rich regions. Whereas here you can see that it is very poor for reads on target. It has variable length target regions, but it also has its own set of advantages. So each of the methods you can observe here, they have some pros and some cons, and none of them are foolproof. But depending on your objective and your requirements, you can choose what is most optimum for you. Next, we will go into details about the library preparation strategy, which is used for WES. Um, First step, if you remember, like we had talked about, the detailed steps of TrueSeq library preparation. It includes fragmentation of DNA, like you previously have also already discussed, which involves the shotgun, which is a part of the shotgun sequencing method. So most commonly for TrueSeq library, what is done is covariance shearing is carried out. Covariance is a very sophisticated ultrasonicator, like I've mentioned previously, and depending on what settings you are utilizing, you can control the length of the fragments which will be generated as a consequence of this shearing. So usually around 150 base pair long double-stranded DNA fragments are generated, and they also have 3' 5' overhangs. Next, what do? Because it's a shearing step, right? DNA is getting randomly fragmented. It's not always going to be blunt-ended in nature. Then what is your next step? You have to repair the ends and you need to select the library sites. Repair ends basically means that you remove those overhangs that have formed as a result of the fragmentation step using covariance shearing, and then you convert them into blunt ends using an end repair mix called as ZRP3. Again, this is a company specific nomenclature. It's sort of the jargon that they use, so we just go along with it. And within this repair mix, you also have a 3' to 5' exonuclease activity, which removes 3' overhangs, and 5' 3' polymerase activity, which fills in the 5' overhangs. So just to quickly, just a second. Yeah. So if you remember, like for any sequences which are double stranded in this manner, you will have a sort of 5' end and you will also have a 3' end, right? So for each sequence, this is the 5' and this is the 3', and here this will be the 5' and this will be the 3'. So whenever this sequence is undergoing shearing, it can have random cuts, right, in this manner. For example, in random breakage points. So as a result of this breakage, what you will eventually end up having as a consequence of this fragmentation in a large number of cases is that you will have certain regions like this, which are double stranded, and what is the result here? You will have a 3' overhang in this case, because here it is 5' to 3'. Also, what you can have is that here you have a 5' to 3' sequence, and therefore what you have here is a 3' OH end which is empty or which is extended. Now, whenever you have an enzyme which is able to, okay, let me just change the color. Yeah. So whenever you have an enzyme that is able to sort of repair these ends, what does it do? It ends up give me a second. I think I did the same thing here. Yeah, sorry. Let me correct it. Um, this I mislabeled. So what you had here, yeah, what you had here was a 3' to 5' fragment. So what you this is 3' to 5', and this is 5' to 3'. Right? Now, from this available 3' OH, what an enzyme or what a polymerase can effectively end up doing is that it can synthesize this region and it can fill in these gaps, finally generating a complete double-stranded blunt DNA end. Or what it can instead do is that it can end up the use of an enzyme which is able to chew up, or an exonuclease enzyme which is able to chew up any 3' overhangs. What is that going to do? That is going to completely remove, okay, sorry, but yeah, that is going to remove that region, and it is going to also end up generating a clean blunt DNA end. Now, once that DNA end has been generated, what will then be done? You will end up repairing the end, and you will end up also generating the library size in this step. That is done using sample purification beads, which will select for fragments of only a certain length. Again, you can read up more about this using the manual that is provided on the Illumina website for the FluSec library or any of the libraries that you want to learn about. Next crucial step is adenylation of the 3' end. Now, somebody might ask why. The reason for this is that a single A nucleotide or adenylation at the 3' end is carried out of these blunt fragments to prevent them from ligating to each other during the adapter ligation reaction, because there a ligase is present. So a continuous stretch of 3' OH ends can actually ligate with each other, can actually ligate. Sorry for the interruption. So, what actually happens here is that you can see that the library size can be selected, and following that, an adenylation step is carried out to prevent any incorrect or any random ligation from proceeding. A corresponding T nucleotide, which is present on the 3' end of the adapter, also provides a complementary overhang for ligating the adapter to the fragment. What does that do? That basically prevents the formation of concatenated templates. If you remember, we had seen this in the previous class when we were talking about long read sequencing. Oh no, DNA nanoball generation, when we talked about that, where four adapters were ligated and a continuous stretch of um, templates could be generated, which can then be subjected to sequencing. So that was a different purpose. But when your objective is not to proceed via that or to, and instead to go for um, bridge amplification by the Illumina strategy, then that becomes a challenge. And so you don't want that to proceed. Um, Following this, you ligate your adapters. You ligate multiple indexing adapters to the ends of DNA fragments, which prepares them for hybridization onto a flow cell. Then you enrich for DNA fragments using PCR to select for only those DNA fragments that have adapter ligated. And then you amplify the amount of DNA which is present in your library. PCR is performed with a PCR primer cocktail that anneals to the ends of these adapters, and you minimize the number of cycles so that you don't um, misrepresent the library in any manner. This is again a very crucial step, and how the companies control it is not something we are discussing today. And a lot of that information is actually not available. Um, Fragments that have no adapters will not be able to hybridize to surface-bound primers in the flow cell. Fragments with an adapter on only one end will be able to hybridize to surface-bound primers, but they will not be able to form clusters. So basically, you will enrich for only those fragments that have adapters on both ends, and that will only those will be subjected to sequencing in the subsequent steps. Then you need to validate the library. You need to quantify them using qubit, for example, and you need to check the quality of the library using a bioanalyzer. All of these steps are, of course, they are a part of the QC step basically after checking the DNA quality. That this library preparation has been performed perfectly and is reliable, because the it's sort of a preliminary check to make sure that whatever you are using for the sequencing process is already of good quality, not contaminated by salts or not unreliable in any manner, and can be used for sequencing, and the data which you get from there is also reliable and can be used in subsequent steps. um yeah Hybridized probes. This step, it actually enables us to combine DNA libraries that contain unique indices or indexes into a single pool, and then it binds targeted regions of the DNA with capture probes. So you have generated your library, you combine multiple libraries which all contain unique indexes into one pool, and then they are able to bind to capture probes to some specific regions. Again, the probes we have discussed already, they will select out for the exonic regions of the genome. And each of these kits, they have different types of probes which are able to recognize exons to a certain extent, depending upon which library they are a part of. Then you capture these hybridized probes using some streptavidin magnetic beads. You you also subject them to repeated washers, heated washers, which removes any non-specific binding. And then you elute out the enriched library from the beads and prepare them for a second round of hybridization. For second hybridization, this step binds targeted regions with capture probes a second time. So you are basically selecting out any non-specific binding or any random binding, and you are increasing the specificity of your captured regions to a larger extent. And in this, this can also be combined with the use of SMBs or streptavidin magnetic beads to capture the probes hybridized to the regions of interest and remove non-specific bindings, and then elute the enriched library and prepare it for sequencing. Finally, you clean up and amplify the captured library. For cleanup, it uses some different sort of beads. So this streptavidin magnetic beads, there are some specific types of beads which allows us to select out the probes, and magnetic beads, of course, help in easier separation. Then, using sample purification beads, you can purify the library before PCR amplification to remove contaminants, and then amplify the enriched library using an 8-cycle PCR program. Again, let me repeat this and stress on this in more detail, that all of this information you can get, like the individual components, the protocol which is used. Of course, it's not completely disclosed. Some things are kept proprietary for the company's own IP, but the manual that is present is going to give you a lot of information. You can read up on it as much as you want, um, especially if it is something that you're going to use as part of your research journey or just for academic proficiency. So, very interesting things, very useful also in the practical sense. So, read up on them. Okay. Next, this is the final slide for today. So, um, I hope that more students join because it's very monotonous otherwise to just keep on lecturing. It's not what is intended. We want interactive topics. I understand that this is new and this is difficult. It might be advanced also for some people, but the idea is to learn new things, not revise old ones. So that's why I'm trying to bring very different aspects for you to learn, from basic biology to commercial sequencing, its application, its analysis, everything. And it's all intended for your good, so that you're more prepared when you eventually venture into this domain. But anyway, I appreciate whatever contribution is has been brought forward from your end. And this is a long course. There is a lot more to do. So I look forward to more participation in the future classes. Anyway, summarizing today's class and the information we have discussed today, you can refer to whatever references I have provided in the slides and any information available online for your own development and own experience. So, from tissue to data, uh, just a second, I think. Yeah, it's fine. So from tissue to data, steps of whole exome sequencing first involve sample processing or pre-processing, which starts with the identification of tumor regions by an experienced pathologist. Um, Followed by DNA extraction, library construction, and amplification. DNA procession commences with a quality check of the reads. If the quality of trimmed reads is sufficient, the alignment of the reads to a reference genome is launched. When binary alignment map or BAM files are processed, so again, this is all information you've not learned about till now, but overall, this is what happens in whole exome sequencing. I just wanted to sum it all up. But basically, first preliminary histopathological analysis is carried out by the lab, the pathology lab that is using that you are um, that is analyzing the sample and looking for tumor regions from the entire tissue, because it's not that the entire tissue is always cancerous or has the disease that you're that you're studying, but instead there are certain regions which are cancerous, and it's always better to get the sequence from that region. From which you extract DNA, then you construct a library, which is what we discussed today, and after amplification, carry out whole exome sequencing. This is where we are. Excuse me. Now, after this, what you get is the data, and that data has to be used for analysis. This is what we'll discuss eventually in our upcoming classes. So as part of data pre-processing, first you have to do a quality control or QC of your reads to check how well they have performed. You need to trim the reads, get rid of unnecessary sequences, which includes adapters and any other indexes, etc., which have been included. You need to map the read to the reference genome, that where exactly has the read come from. You need to check the quality of your mapping. You need to get BAM files, mark duplicates, etc. Check the quality of the bases which are present. And finally, after all of that has been done, you need to do downstream analysis, which includes obtaining information like copy number variation, which is the number of copies which are present for your gene of interest, single nucleotide changes. You can look for, then you can use that information to find whether there are druggable targets which are undergoing mutation in a large number of patients or a subset of patients with a certain genetic background. You can look at intra-tumor heterogeneity, that there are some changes within the same patient's tumor which can lead to more severe disease. Um, Tumor mutation burden or TMB, microsatellite instability, homologous recombination deficiency, all of this can be used to eventually satisfy our goal, which is to find better options for treatment of patients and find druggable targets, which are sometimes you can also opt for drug repurposing, where previously a drug which has been established or which has been studied for one type of a disease can be used for a different type of a disease. So that is something which is also looked at. So yeah, overall, that is what we see here. Um, yeah, I think that is all I wanted to discuss for today. Um, yeah. So let me, yeah, I've stopped sharing. But basically, this has completed our class for today. I look forward to better participation in the upcoming classes so that we can have better discussions and more productive discussions in the long run. And thank you for joining. I'll see you in the next class and we'll have a better discussion then. Thank you.